Yeah. Okay. Good afternoon, everyone. Good afternoon, everyone. And I am thankful to the organizers for inviting me for this event. And the topic that I've selected, I've already communicated, is I'm going to speak on bacterial diversity and biocatalytic potential with respect to saline habitats of coastal Gujarat. That is the theme. Now, the frame of the talk, this is what I'm going to speak on. The nature of saline habitats that we have studied during the last 25 years. Then two aspects that I'm going to discuss diversity of halophilic bacteria on different parameters, and then their enzymes, the characteristics of the enzymes, and profiling of the gene and cloning and expression. These are the aspects that I'm going to discuss in brief, of course, because it's a multiple dimensions. The work that I've included is a vast, and that's why I have to be very precise. Now, as far as the saline habitats are concerned, most of the haloalkaliphilic bacteria, they have been studied from saline habitats, mostly from soda lake, and most of the studies they have confined to diversity and phylogenetic analysis. The rest of the things have not been paid so much attention. For example, regulation of gene expression, how the proteins are folded, what is the exact role of salt. All these things are still not in detailed investigated. Now, these are some of the saline habitats that we have explored over the period of time. Majorly seawater, as we'll see that Gujarat coast is one of the longest coast of India, and seawater is the major saline habitat. Then another is saline soil, and there are a lot of salt making activities along the coast, and that is another habitat, salt pans and nearby areas. And then another is one particular saline habitat, which is saline desert, which is Little Run of Kutch. Now, in this saline desert, most of the studies so far, for the first time, we are doing it non-vegetated, because it's a mixture of dry, wet, and admixture of both. And that is a very unique habitat, unique saline habitat, where depending on the season, depending on the temperature, and depending on the rainfall, the conditions change. So our focus is mainly on the microbial diversity of unvegetated barren soil with respect to Little Run of Kutch. Now, this is just a demonstration location. Coastal Gujarat, among the highest, longest coastline of 1600 kilometers in Indian coast, starting from Dwarka at this end and to Bhavnagar along. So I think more than 1600 kilometers stretch is covered in this, and this is a more precise way. The sampling site, the study sites, the Bhuj, the LRK is one of the key study area that we are going to cover. Then Mithapur, which is a factory, chemical factory, major chemical factory, Tata Chemical Factory, and Porbandar, Verawal, and these are some of the study sites where we have focused our studies. This is a Google map thing, and Little Run. Now, diagrammatically, as I mentioned in the beginning, that it's a admixture of wet, dry, and middle muddy type of, depending on factors. depending on the rain, depending on the dryness, we have all these three habitats represented, and one of the study sites is this habitat, that is saline desert. Now, as far as our research team is concerned, we have divided two different groups over the years. One group is involved in actinomyces, another group involved in single cell bacteria. Now, dimensions of these studies, as I mentioned at the beginning, that we have two dimensions, one diversity aspect and one enzymatic characteristic. In diversity, we have taken all conventional mode, for example, morphological, cultural, cultivation, and biochemical properties, enzymatic profiling, and molecular aspects to judge and assess the diversity. The another dimension is the enzymatic, the distribution of the enzyme, the characteristics of the enzyme, the types of enzyme, and the genetic profiling of that particular enzyme. Now, here I'd like to mention that, however, we have done many enzymes, but I'm going to focus only on proteases, because proteases are the most prevalent and most prominent enzymes found in all these bacteria from saline habitats, and the structure and function relationship of these enzymes. Some representation of the study sites, like Tata Chemicals. This is one of the largest chemical factory producing the table salt, and many other chemicals, of course. Then some other saline habitats along the coast of Gujarat. Now, the different saline habitats, they have their own characteristics, of course, with respect to the salinity, with respect to the microbial diversity, and the salt and pH requirement of those bacteria. So there are different criteria on the basis of which these microorganisms, they can be diversified. Now, one of the major aspects among the microorganisms, as I mentioned, that we have worked on actinomyces, single cell bacteria, as well as archaea. In this presentation, I'm not going to focus on archaea because it will be, it's too much to include it. So I will focus only on actinomyces and single cell bacteria. Now, starting with the actinomyces, as we all are aware, they're gram-positive bacteria with high GC content, and as compared to single cell bacteria, they are more morphologically, they are more advanced. Morphologically, they are more advanced in the sense that the level of morphogenesis is much greater as compared to single. For example, we can differentiate based on their filament, fragmentation, sporulation, type of spores, and so that's why we have much broader avenue as far as the diversification is concerned. Now, actinomyces, as I just mentioned, that mycelial structure, the sporulation, the time required for their sporulation, the morphological features of the spores, these are the criteria to judge and to diversify the actinomyces, and that's what we have done for these organisms from saline habitats. Now, those who are familiar with actinomyces, they will be able to see that actinomyces, they have very typical look and appearance on the petri plate. As you can see here, that typical dryness in the appearance, a powdery look in the appearance, of course, a typical smell of this, which is characteristic feature of actinomyces. Now, another feature I would like to highlight here is these are representative diagrams showing that certain features of the actinomyces is dependent on the nature of the medium. As it's shown that the actinomyces isolated from seawater and using different media, same isolates grown on different media, they have different features, particularly in terms of the pigmentation, and that leads to the change in color. And you can see that on different media, that different color appeared on that. So the changing nature of the pigmentation leading to change in color and overall quality. Okay. Now, morphological effects. The light microscope, and most of the actinomyces, they are filamentous in nature, the thin thread-like structures, but some of them, when they are fragmented, we can see this kind of picture here. Yeah. Now then, some of the features reflected in electron microscope, ACM, which we can see that salt crystals can be visualized along with the fragments. Yet another example of actinomyces from seawater. Mostly they are filamentous in nature, and depending on the stage of growth, some are fragmented and some are in sporulating states. Now, another thing that that I'm going to focus is the in situ culture technique on slide. We can observe the changes in the morphological features. We can see the germination of the spores, fragmentation, and leading to formation of spores, and germination and fragmentation, so this profiling can be followed under in situ condition in real time in slide culture, and that is a very useful way to differentiate different actinomyces. They may look similar, they may look same thread-like structure, but there are many features on the basis of which they are quite differentiated. Still, colony morphological features in bacteria, there are very limited ways of differentiation, but we can see here that based on the conventional features of the colony, for example, shape of the colony, texture, elevation, margin, the differentiation can be done, and the organisms isolated from different seasons, winter, summer, and monsoon, can be diversified based on these conventional features. The same thing reflected for the bacteria isolated from saline desert, the little run-up saline desert, which is a mixture of dry land and wetland. Morphological features of the colony can be used effectively to differentiate and diversify the organisms isolated from different habitats. So, morphological features, colony morphology, then biochemical features, biochemical fingerprinting, certain biochemical features, for example, production of enzymes, urea utilization, and production of F2S and indole. These are some of the features which can be used, that different microorganisms have different responses, presence or absence, and also the level and intensity of these features can vary, and based on that, profiling has been generated. And these are some examples. Similarly, for the sugar utilization profiling, the ability of the organism to use a range of sugars, and that can provide a way to diversify them. Now, still, this is just the same theme reflected, that the marine actinomyces from seawater, the metabolic characterization, the actinobacteria from winter season, summer season, monsoon season, they reflect different profiling based on these biochemical features. The same thing is reflected from those bacteria from another saline habitat, that is saline desert. Now, another feature is antibiotic resistance and sensitivity. Antibiotic resistance and sensitivity among the organism is very useful tool to diversify them. And as some of the examples I would like to show, antibiotic resistance plus antimicrobial activity. These are the two features that these actinomyces can inhibit the growth of certain microorganisms. So antimicrobial activity as well as antibiotic resistance. This is just some examples that different microorganisms have different profiling against different antibiotic resistance. Now, we can generate secondary data based on the functionality. For the different antibiotics have different mode of action, and different mode of actions can be used for the generation of secondary data. For example, functional means, so this is the secondary data which is generated, and based on this secondary data also we can generate a profiling and diversify. Now, the another feature is related to the antibiotic resistance is multiple antibiotic resistance index, and based on this index, actually this index is a parameter to reflect the intensity of the resistance character, that is for one single organism or a particular organism, how many antibiotics it's resistant against. So that is the reflection. High number means large number of antibiotics is able to resist. This is another quantitative parameter based on which actinomyces have been characterized or diversified rather. Now, molecular approaches. To study the actinomyces, they include mostly the basis is 16S ribosomal RNA gene, and there are two dimensions. Using 16S, of course, based on universal primers, we can identify as well as we can generate profiling for their phylogenetic relatedness. The another aspect that we have attempted is using multiple primers, some universal primers, some organism-based primers of 16S. We can generate profiling of the amplification, and using that profiling of amplification, we can differentiate organisms from different habitats. We'll show some examples. Now, briefly, in addition to 16S molecular parameter, there is still another parameter, chemotaxonomic parameters. For example, cell wall contents, sugar content and amino acid content. We extracted the cell wall sugars and cell wall amino acids. Based on sugar contents and amino acid contents, the organisms have been diversified. Now, coming back to 16S, we can see that based on different primers, 5 different primers, and different organisms, means DNA isolated from those organisms can be amplified, and amplification profiling using these primers can be very useful parameters, as we can see. And of course, for that we need to establish the PCR conditions with respect to the particularly the annealing temperature. And for example, this profiling is for 2 different primers. The universal primer for actinomyces, this one, and universal primer for the another universal primer for this. Then another set of primer, organism-based primers, and using the amplification profiling, we can generate this spectrum. And as you can see, that this upper panel is for one particular habitat, the lower panel is for another habitat, and we can see the amplification profiling of these 5 different primers. We can see the difference, and this can be a good basis to differentiate organisms from one site to another site. Then, of course, amplified ribosomal DNA ADRA that can, using certain restriction nucleases, we have generated profiling and converted into tentogram, which is another way of diversifying and these organisms. Now, phylogenetic analysis, this is just a representation that actinobacteria isolated from winter season, monsoon season, and summer season. As this reflects that the phylogenetic relatedness is quite distinct. Seasonal variation, seasonal distinctness is revealed in this phylogenetic related of actinobacteria. Now, same distinctness or same diversification is also reflected. Similar diversification is reflected by another habitat, and that is saline habitat LRK, and the bacteria generated the bacteria from 3 different habitats of LRK, 3 different habitats, and these study locations, they are separated by 50 to 100 kilometers away. So 3 study sites, Surajbari, Venasar, and Jogat. You can see the organisms. Some organisms they are common as reflected here, but they have distinct genre-wise profiling as reflected in this case. Now, another point is phylogenetic relatedness and Phenogram means the phenotypic parameters. We can generate phonograms based on phenotypic characters, and similar to phylograms, profiling can be generated. But if you compare these two approaches do not necessarily correspond each other. There are variations, means the clustering pattern can be different. Clustering pattern reflected in phonograms can be different as phylograms. So these are some examples, like these two organisms, they are placed together, whereas they are placed far apart. Now, novel lineages have been found in some cases, and they have been reported based on unique profiling, lipid profiling, fatty acid profiling, and other parameters. More recently, this is we studied this particular bacteria, and we found that this is a different. This is a distinct from the Bacillus subtilis, the type, and there's a Bacillus alkali-philus, and that is recently published a few years ago. Now then, enzymes. The organisms, they produce different enzymes. Proteases being the particular, and the enzymes have certain key features. These enzymes have key features. For example, wide occurrence, pH and salt profiling. There are certain features, for example, salt-dependent stability, and the effect of salt on their expression. These are some of the common features which we have noticed for the enzymes obtained from different saline habitats. Now, these slides actually, they represent different sites of saline habitats, the different organisms isolated from different sites, the beta-2, DIU, and the enzymatic profiling. Again, the same spectrum, different sites, different organisms, and different enzymatic profiling. Now, one segment of this presentation actually includes actinomyces from seawater, and as part of the Minister of Earth Sciences, Government of India, we studied actinomyces from two different seawater, one pristine seawater and one polluted seawater, and we compared the diversity and enzymatic profiling in different seasons. For example, pre-monsoon, monsoon, and post-monsoon seasons. And this is one particular pristine habitat, and the another profiling is from more polluted or more heavily humanly infested, with more activity. For example, ship breaking yard, that is one of the major ship breaking yard in Alang. So the profiling of these two sites have been compared with respect to the diversity, with respect to the enzymes they produce, with respect to the seasonal diversity, and this is what reflected with these slides. Now, another thing which I'd like to mention here is that the level of enzyme production. The level of enzyme productions among the organisms isolated from different saline habitats, it varies. For example, this is the level of protease production in the bacteria isolated from seawater, and there are comparable level of activity. Whereas if from different habitats, for example, salt pans, the organisms isolated from salt pan, they have different profiling. The variation in the enzyme activity is quite huge as compared to previously. Then there is a variability with respect to their activity, stability, the temperature profiling, pH profiling, and salt range and requirement. Now, this is just to sum it. Most of the organisms, they are able to grow in the presence, optimally grow in the presence of 5 to 10% salt, as well as produce the enzymes optimally. Similarly, in the pH range of 9, most of the organisms, they are able to grow in pH 9 optimally and produce the enzyme. Similar pattern is reflected in marine actinobacteria from seawater for recent studies. Now, another feature is related to production, that there is the repression. With some studies we have focused on the repression, that is synthesis of the enzyme, which is governed by carbon-nitrogen ratio. And we have seen that these actinomyces, they are able to grow in minimal medium with amino acid as both carbon and nitrogen source, and they can grow, they can produce, but the level of amino acids, the combination of amino acid is a threshold. Okay, as it's mentioned here, that up to certain level or combination, it induces the enzyme production, but beyond which it represses the enzyme production. And this is what it's reflected here. Now, purification and characteristic features of the proteases. Purification and characteristic features of the proteases. And purification has been a tricky because salt requirement, so we have to devise those methods which do not entirely depend on the salt, because the moment you remove the salt, the enzyme becomes unstable. So some of the common features of the enzymes, particularly when I say enzyme, as I mentioned in the beginning, I mean proteases, because we have focused in this presentation on proteases. In general, the proteases from saline habitats, they have optimum temperature at 60 to 70 degrees. That is very interesting, because organisms are going under mesophilic conditions, but their enzymes is quite stable. Not only the stable, but it's optimally active at 60 to 70 degrees, as well as pH 9. Now, some other features related to the one key feature is that salt-dependent shifting of the profiling. As we can see that in the absence of salt, the optimum temperature is somewhere around 50 or so. And this can shift. In the presence of salt, there's a shifting, a significant shift in the temperature profiling. The optimum temperature shifts towards higher temperature, and this is reflected in many organisms from saline habitats. This is another example. The same pattern is reflected. And another example, we can have the same pattern, that in the presence of salt, there is a shifting of the temperature profiling. Now, there is a variability with respect to salt requirement for catalysis. The salt required for the stability is different. Salt required for the catalysis is different. Here we are showing that salt required for the enzyme catalysis. Many other factors which are required. Most of the enzymes, they are highly active in surfactants, and that's the usefulness of these enzymes. Now, another feature of the enzyme is their resistance against thermal denaturation. Thermal denaturation and chemical denaturation. Sorry, that most of these enzymes, they resist urea denaturation. But we found that this resistance character actually is due to salt, because when you dialyze, remove the salt, then it becomes highly sensitive. So this ability to resist urea denaturation is due to salt. Now, thermodynamic parameters have been studied, and it has been shown that in general these enzymes have negatively charged, and the purpose of negative charge, or the consequence of negative charge, is that the hydrated group is shielded, and because hydrated group is shielded, so it's a kind of protection, and due to this protection, the enzyme remains in soluble state. So in the presence of salt, enzyme is soluble, and if you remove the salt, then enzyme becomes precipitated, and it will lose the activity. And the similar parameters and the parameters have been studied, for example, thermodynamics, entropy, enthalpy, and free energy. The catalysis KCAT, the catalysis this catalytic constant KCAT is reflected in this case, the correlation between temperature and salt. The enzyme loses its catalytic efficiency when you increase the temperature. But this process can be reverted, this can be retarded in the presence of salt. As we can see that when there is no salt at 80 degrees, almost enzyme has lost catalytic efficiency, but in the presence of salt, it is significantly protected. Structure and function studies have been done with respect to internal analysis, with respect to the structural stability and the bonds which are responsible for the stability. The secondary structures have been followed by CD, mostly FTIR, and because of the time limitation, I am unable to go, and I think I have 5-10 minutes left. How much time I am left? Is it okay, madam? Is it okay if I get 10 minutes?
Yes, 10 minutes left.
Okay, thank you.
Welcome.
Now, with respect to structural stability, I would like to highlight this particular study that where we have immobilized the enzymes. We have immobilized the enzyme using different approaches and large number of carriers, and structural elucidation of the immobilized enzymes have been carried out. And as it is reflected here, that structural topography of the immobilized enzyme, and it has been found that amide A band is very important, and in this interaction amino group plays important role rather than carboxylic group, as revealed by FTR analysis. Now, another feature of the enzyme is non-aqueous. Most of the enzymes are able to function up to a certain extent in the presence of solvents, and more recent studies as reflected here, that we have compared 5 different represents of enzyme, 2 native enzyme, 2 recombinant enzymes, and 1 metagenetic derived enzyme, and all these enzymes, they have different responses with respect to their solvent tolerance. They are able to function, but at different efficiency. So this is a more recent outcome of our research. Then cloning expression, the gene number of enzymes have been cloned in mostly in E. coli, but before cloning we have also done profiling of the protease gene, and profiling has been done using different primers. Primers have been synthesized. Some of the primers are bioinformatically designed primers. Some of the primers are based on the organism. Some of the primers are based on known sequences already published. So using those primers, we have generated a profiling of protease gene, and then we compared from different saline habitats. One particular enzyme which is recently gene cloned from saline habitat, saline desert, and published 2 years ago in Frontiers in Microbiology, and the structural elucidation has been done. The factors affecting expression has been done. The growth temperature, the induction, and also the solubility. These are the things that we studied with respect to expression and effect of salt. How salt affects the expression. And this is just one example how we can enhance the solubility, because in overexpression vectors, mostly enzymes go into the inclusion bodies. So just to prevent that, we can optimize the level of growth, the level of induction, and also some other factors. Perhaps you can use the molecular chaperones. So these are some of the conditions which can be used to prevent the formation of inclusion bodies. And then comparison of the recombinant enzymes with the enzymes producing their native host can be compared those produced in E. coli. Primarily, there is not vast difference, but in some cases there is a difference. For example, few degree temperature difference, and otherwise, broadly speaking, they are similar when producing different hosts. This is the example that I mentioned, that the structural elucidation of one of the enzymes, this derived 3D structures, N-terminal and C-terminal, the enzyme which is derived from bacteria from saline desert. And these are some of the features, that 337 amino acids, the conditions for the optimum expression, and the as 8 familiar proteases. So these are some of the features have been elucidated. Now, the stability of the sequences have been that most of the amino acids, as reflected in Ramchandran plot, is in the most favored region. Now, metagenomics, some of the enzymes were derived from metagenomics, and before for this study, first thing is that we need to establish the conditions for the extraction of the metagenome. So from different saline habitats, we have established the conditions for metagenic extraction, and using this metagenome, we can go for studies related to their diversity, related to creation of metagenomic library, and also creation and also search for new genes. So these are some examples where you can see different saline habitats reflect differently in terms of their phylum-wise diversification, genera-wise differentiation, and also in terms of their OTUs and the abundance that has been studied with respect to saline habitat. Functionality using PyCrust software and case pathway. Most of these enzymes, most of these habitats, they have reflected diversity in their metabolism and their ability to utilize hydrocarbons. So that is one of the useful features. It can be used for bioremediation. Now, to conclude my talk, wide occurrence of the enzymes which are present in the microorganisms from saline habitats. There is a variation with respect to pH range, temperature range, salt range, and there are certain key features, for example, salt-dependent thermostability and salt-dependent resistance against chemical denaturation. These are the common features which emerge. Now, with particular references to Little Run of Kutch, and the first study which have been extensively studied in such a habitat which is low in nutrient level, because we have taken dry areas, unvegetated area, very low rainfall, low nutrient content, and we have studied the microbial diversity in that region. And isolation strategies we have adopted, we have modified the isolation strategies using different factors, and considerable phenotypic variations have been observed, and majority have produced enzymes, and majority of the organisms have reflected cosmopolitan distribution. Now, with respect to seawater, nutritional variability and ability to utilize its a versatility. Organisms are highly versatile in terms of their nutrient uptake, in terms of their sensitivity and resistance against antibiotic, and also in richness and abundance. With respect to metagenomics, protocols for metagenomic DNA extractions have been established. Then polluted and pristine studies have been compared. Then one polluted side, one pristine side compared, and microbial diversity of these two have been compared. And mostly we have found that most of the organisms, they have been identified as unclassified. And many novel lineages, there is some we have studied, but there are prospect of many other novel lineages, and of course genes and pathways which can be further investigated. This is the time to acknowledge our research group, and large number of PhD students, 25 PhD students who have worked over the period of 25 years. The work that I have summarized actually is a summary of the work which has been based on work done by these students. 25 PhD students, 4 PhD students are still working. And the financial support from funding agencies, DBT, UGC, DST, Ministry of Earth Sciences, GSBTM, and our own university, our research collaborations. I would like to acknowledge Professor Khare from IIT Delhi, Professor Sanjay Kapoor from Delhi University South Campus, Professor Kiyoshi Hayashi from Tsukuba Science City. Now he's with Toyo University, Griffith University, Australia, and Jawaharlal Nehru Technical University in Hyderabad, and Central University in Hyderabad. There's a functional collaborations with which we have, with them we have worked together. We have joint publications and we have joint research projects. These are research publications. Some of the recent research publications, which we have also shown during the presentation, edited books. A few years ago, we edited this book on microbiome, and large number of, I think 5 book chapters that we have contributed to different edited books. Few databases we have created. This is our research team, past research team, current research team, and still 4 PhD students and 4 or 5 master students who are working on this. I thank you once again. I hope that I have maintained the time limit.